Welcome back. For some reason, muscle development. This slide, I don't know why that does that. The other thing, this is the second time today. I need to work on my posture. So tall here. The second thing, this is the second lecture that I forgot the record button didn't hit. I'm wondering if there's an issue with if it's being weird, because usually I never do this. Luckily, this is a very short lecture, so I'm not as perturbed as the last one. Anne Frank. Not Anne Frank. I've been so bad with my verbal slip-ups. Lisa Frank, the other Frank, is who this art style, very 90s. I don't know if it ever came back. I forget which, some students, I had a student come to office hours the other day, and she was wearing kind of bell bottoms or whatever you consider that style now. And she was waiting outside, and I said, I used to have jeans just like that when I was in high school, which probably mortified her. Jinko jeans, that was the thing back in the 90s. But every 20 years, it seems like bell bottoms comes back. Anyways, so I don't know, bringing that back to Lisa Frank, not Anne Frank, I don't know if she ever came back, if she became popular again, or this art style, the kind of poppy unicorn rainbow art style came back again when you guys were young or not. But anyways, or if it's popular now, who knows. Let's move on. This is what happens when I record a lecture and then it doesn't record. Then I just get loopy about it. Let's talk muscle development. So we have our paracrine factors from the notochord and the neural tube. They are influencing those myotome cells in the dermamyotome, right? They cause determination. Those paracrine factors, WNT, sonic hedgehog, MyoD, myo anything is muscle, right? And then MyF5, what they do is they cause these myoblasts to start multiplying and dividing. FGFs are then helping the transition to the cell alignment stage, where we have multiplication of the cell stop. Fibronectin, integrin, cadherin, CAM, and myogen cause them to align like this. We have the initial myotube formation, where we get fusion of these cells. So you can see that one cell's got multiple nuclei because these have all fused their cell membranes together. Then we have meltrin muscle and meltrin specific proteins involved in this process. We have the myotube maturation, so it's starting to fuse more. So we have beginning of the myotube fusion. We're getting more fusion here. Most of the remaining myoblasts are fused in. Interleukin 4 is involved here. Then lastly, we have muscle fiber and stem cell generation and maturation, where contractions begin. Muscle contractions, not like pregnancy contractions, muscle contractions. So let's talk, so that's kind of the development of these muscle fibers, but we also have these things called satellite cells. And this is important for muscle growth. So if you're, lifting weights to build your guns, get your tickets to the gun show, these are the important players, the satellite cells. So this one here has been stained with PAX7 to make it glow. We talked about PAX, some of the PAX genes before, and the inheriting the PAX gene kind of when you have this asymmetric cell division. If you inherit PAX in your cell membrane, you become one type of cell. If you don't have as much PAX, you become a different type of cell. Very similar mechanism here. So here we show this asymmetric cell division of a satellite cell. And so the red cell at the top here is keeping that PAX7. It has more of the PAX7. In the daughter cell, it's expressing MYF5. That differentiates these two, where the PAX7 one still remains a stem cell, so it can then either undergo asymmetrical cell division itself to make more stem cells. The bottom one here It has that MyF5 and becomes a progenitor cell, so it will eventually become muscle. But that can undergo cell division as well, and we'll get into that in a second. All right, so here's kind of a little clearer picture and not a immunostained one. So we have our stem cell that is bordering the myofiber, so that's your normal muscle tissue. It can undergo cell division, and the stem cell keeps PAX. The progenitor keeps MyF5, right? So you have this will eventually become more muscle tissue. The stem cell remains a stem cell and just waiting to generate more tissue. When you start lifting those, doing curls for the girls, I don't know what all these weightlifting terms are. That's what does it. So basal lamina. We have here, you can also have, so that was asymmetrical cell division. Let me step back. We have asymmetrical cell division where one progenitor cell or one stem cell becomes another stem cell and a progenitor cell, but you can have symmetrical division too of both. So you can have a stem cell or a progenitor cell that undergoes cell division, and you can duplicate, you can have two stem cells then, or you can have two progenitor cells then. All these mechanisms work. So a stem cell can become a stem cell and a progenitor cell. A stem cell can become a stem cell and a stem cell. A progenitor cell, after it came from a stem cell, can become another progenitor cell. So that's it. That's it. That's the lecture. 7 minutes. Boom. It's pretty straightforward. I mean, it's not like we're not super in depth, but muscle stimulation. So what you're doing is when you're lifting these cells, a lot of times you're like kind of tearing or putting a lot of mechanical stress on your muscle tissue, right? When you're lifting weights or running or doing crunches or whatever exercise you're doing that causes stress on your muscles. And what happens then is that stimulation is recognized. These stem cells or progenitor cells say, oh no, I need to start replicating. Like, this stress is not attainable to a long, successful organism. And so we're going to replicate, devote resources to these muscle cells to be able to cope with this stress or this required output of the muscle tissue. And so that leads to this either symmetrical, asymmetrical replication of these cells and the building of muscle tissue. So that's kind of the synopsis there. All right, that's it. Easy breezy muscle tissue. Muscle tissue is pretty basic. Like, there's not a lot of differentiation. It's just like we just need to make more. If you have a lot of stress and you need to lift a lot of weight, they don't have to like change how the muscle fibers work or anything. They just need to make more of it. And that's why you see people get swole or whatever. It's like you get bigger muscles if you need to lift more weight. So that's it. I hope you enjoyed the lecture, and I'll catch you in the next one.